wondered about sex? I mean, why we have it? Because when you think about it, it doesn't make a whole lot of sense. I mean, Darwinian sense, survival of the fittest and all that. And, well, why would you combine your genetic material with somebody else's? Why would, well, I've checked this audience out. Most of you aren't going to bear the next generation, so what's the benefit of all of this? And what makes something like this fit? Or this? This is really kind of a big question for me. I think about this a lot. And the reason I think about this is that I am, as you heard, I'm both a scientist and an engineer, and I'm really interested in building new biological objects. I like to be inspired by the beauty and the functionality of the natural world, because when you think about it, here the natural world has solved an immense array of problems, such as extracting materials and chemicals and energy from the environment, right? And converting that into remarkable, self-repairing, self-replicating machines. These are the things that have solved the problem of life over and over again. And for me as an engineer, that's an incredible inspiration, because when it comes to making beautiful and functional things, nature is the best engineer there is. Humans can't compare to the products of 4 billion years of evolution. Okay, so what does this have to do with sex? Let's get back to the topic. Sex is important because, remember, all of this functionality of nature is encoded in a simple, elegant polymer, a 4-letter string. A 4-letter code dictates whether you're a microbe or whether you're a monkey. And you can synthesize virtually any DNA you want, right? So the technology, actually think about this, the technology has gotten to the point where you can synthesize anything. You can type out a sequence on your keyboard, email that sequence of DNA to your favorite house in, DNA synthesis house in Taiwan or South Africa or Northern California, you name it. And in a few weeks, you will get back in the mail your genetic material, a gene. I mean, this is really quite amazing that you can basically synthesize anything you want. In fact, many in this crowd will remember the headlines from a couple years ago where Craig Venter's gang synthesized not just a gene, but an entire genome. And not only did they do that, they put that genome, basically made from non-living material in a test tube. They put that back into a cell, a bacterium, a rather dull bacterium, but a living bacterium nonetheless, or wasn't. This is the whole question, was it alive? They put it back into a bacterium whose DNA had been removed, and that cell rebooted. It read that DNA as its own, and it started following those instructions and doing what that DNA told it to do, started reproducing. That's quite a remarkable feat. But here's the problem, from my point of view, because I want to build new biological things. I want to write new DNA. Is that who wrote that music, right? They synthesized the DNA, the cells read the DNA, but they didn't compose that genome. Because when you think about it, the DNA, the code of life, is like a Beethoven symphony. It's elegant. It's stunningly beautiful. And we don't know how to write music like that. What we're doing now is basically we're cutting and pasting passages from nature's symphonies, maybe rearranging them a little bit, but we're not writing it. So you think TED Talks are short and sweet, much shorter than the usual talks that I give. But how about a nano lecture? You ever heard of a nano lecture? One of the highlights of the Ig Nobel Prize ceremonies is a lecture delivered by an eminent scientist, limited to 24 seconds. And Eric Lander of the Human Genome Project did an absolutely brilliant nano lecture in 2003, which I will repeat here because it describes so well how difficult it is to make sense of the mysterious workings of the genome. Human Genome Project, biology's moonshot. 15 years, 6 countries, 20 centers. $3 billion, 3 billion letters. $1 a letter, such a deal. 23 chromosomes, supposed to contain 100,000 genes. Turns out only to have 30,000 genes. Or maybe 25,000, but it could be 40,000. Check back next year. Said to have the answer to absolutely everything. Absolutely everything. Diabetes, asthma, cancer, evolution, populations, migrations, life, death, taxes. Even the Boston Red Sox. The only problem is there's no index. That pretty much sums it up, 24 seconds. But even better, for this ceremony, you have to have an abstract. And your abstract is 7 words. All right, so use that as your own challenge. How would you describe your field, your whole thing in 7 words or less? His was great. Genome. Bought the book, hard to read. Well, I have to admit that one of my longest-held ambitions has been to be invited to give an Ig Nobel Prize lecture. I have not been invited. I'm not sure I want to win the prize itself, but the prize lecture is a good one. I've even prepared my own abstract, because when I come to this problem, I like 6 words, 7 words is about all I can handle. So here's my abstract. Genome, great story. Hard to write. Count the words, 6. That means I have 1 left. That's because I'm leaving space for sex. The answer is sex. The answer is at least evolution. Because who knows how to write even a single protein? A protein that would do something useful for humans. I think that biology can solve human problems. Coming back to what I want to do as an engineer. And nobody knows how to write out a single protein that would, say, cure cancer. But proteins are not designed. In fact, they are evolved. They are the products of Darwinian exploration, innovation, trial and error, success and failure for billions of years. And that has created an immense array of molecules that can do amazing things. What is a protein? It's encoded by the DNA. The DNA encodes the proteins. Proteins do just about everything else. They collect everything or make everything that your body needs to survive. They convert sunlight into chemical engineering energy. They convert chemical engineer energy into motion. They do about everything that you need in order to survive and reproduce. So to make new ones, and to use this process of evolution, that's what I do over there in California. I have fun forcing molecules to have sex. And with that, we can create things that don't exist in nature, right? Because think about sex. Sex in nature is pretty limited. Dogs go with dogs, frogs with frogs, monkey with monkeys. Always 2 parents, limited to just 2 parents. Always limited to within species, but this is not the case in the test tube. In the test tube, remember, we can synthesize anything now. The DNA can be anything that I can write into my keyboard and send off. So what do I write? The process has now become wide open, and when it's wide open, your imagination can take you where you want to go, or your curiosity. So what is it that we make? How do you make something that's wonderful? When you start having control over the process like I have, you can mix 2 parents or 32 parents. You can cross a monkey gene with a worm gene. But the problem is that most of the ways that you can do that don't make anything at all, anything useful at least. And so the question becomes for me, how do you actually do this in a way to make something wonderful? All right, so let me just show you briefly what I'm talking about, right? It's easy to say, oh yeah, I force molecules to have sex, but unfortunately, this is not as much fun as some of you might think. It all happens in the test tube. There's no romantic music or anything. But I have a lot of control over what happens. And it looks a little bit like this. A protein, as you know, is a three-dimensional object that's encoded by a linear string of DNA letters. And this three-dimensional object has, if it's related to another protein through evolution, right? Somebody a long time ago discovered a protein. They accumulated mutations over millions of years. If I now take these proteins from distant species and I breed them together, what I'm doing is I'm taking their DNA and cutting it into pieces, either literally or figuratively, and assembling those pieces. And this works because the proteins keep a similar three-dimensional structure even over very long periods of evolutionary time. Now, what happens then is that when I cut these into pieces, I choose. I'm the one who's going to choose how to cut these into pieces. And I'm going to shuffle these pieces together again to create progeny sequences. And I can make lots and lots of progeny sequences this way. And because they came from related molecules, they will, to a large extent, especially the way that I particularly choose to do this, reassemble into functional three-dimensional protein structures. What's the safe sex part about this? Well, this is kind of the interesting piece, because with the control over the process comes this need to create something useful in a very short period of time. That's really the challenge. Evolution has billions of years to create things, throws most of them away, or many of the changes away, and can collect over billions of years. My grad students don't have billions of years in order to be successful. So we have to contaminate our blind experiments with a little bit of thought. We have to make choices on how to cut these pieces of DNA and reassemble them. So we ask questions like, what are the sub-elements of a gene that when swapped among parents will contribute to a three-dimensional structure, a functional protein? And we find that we can apply rules, for example, that computer science stole from genetics in the first place, genetic algorithms, and put them back into the breeding of proteins. Thou shalt break the fewest interactions, for example, so that we can recombine things and have a chance, even if they come from very distant species, of making a molecule that works again. And what do we get when we do this? Well, we get really interesting things. First of all, we see when we breed proteins from distant species that, and especially when we do it my way, that we get proteins. We get functional proteins that you can breed DNA from very distant species, and you get things, just like when you breed cats, you get 2 ears and a tail and 4 legs, usually. We find that we can get the same sorts of things, that it's a conservative process, right? You can make lots of mutations, but do it in a way that conserves the overall structure and function of the molecule. But what makes all this worthwhile? Why is that interesting? Well, that's not the most interesting part. The most interesting part is we find that sex is an innovation generation machine. Because when you think about all the children that you could make, right? Some look like their parents, some are dummies, but some are much smarter than their parents, and some can do things that their parents cannot do. And that's just when you breed humans. Imagine when you're breeding molecules from lots of different places. So we find that we can make really interesting and useful things. For example, arginases. These are enzymes that scavenge a nutrient from certain tumors, arginine, and have been considered as cancer therapeutics. When we breed 2 human versions of this enzyme together, we can make molecules that last much longer in the bloodstream than any of their parents. and therefore could be used as a much better therapeutic agent than the naturally occurring enzymes. And because they come from humans, we think that the children or the progeny sequences wouldn't elicit an immune response. We've been able to take bacterial enzymes that you can literally scrape off the bottom of your shoe and turn those into mimics of the human liver. Cytochrome P450s, remember your first line of defense in xenobiotics. These break down drugs in the body. We can take these bacterial enzymes and turn them into the human equivalents so that you can make large amounts of the actual human metabolites of drugs and test their toxicity and their biological efficacy before you even take a drug to clinical trial. A very important application of this technology has been in learning to live sustainably, in converting new and useful sources of energy like the sun into liquid fuels that we need, instead of pulling our riches out of the ground as we've been doing for the last 50, 60 years. Biology is great at this, and we've been able to make enzymes, for example, that extract the sugars from biomass and grasses in order to convert that into liquid fuels. It's really been a fun exploration when you think about it. Evolution is a very powerful algorithm at all levels of complexity in biology. And when you don't understand what it is you're trying to make, it's an extremely good algorithm for forward evolution. So I look forward to future exploration and future experiments in this, and it's been a great deal of fun. Thank you for listening.